Ulrich Massing. He's a chemist by training. He received his PhD in biophysical chemistry from the Max Planck Institute in Göttingen, and he's now professor of pharmaceutical science at the University of Freiburg. Please, the stage is yours.
Yeah, Simon, many thanks. Is this micro I have to need? Yes. Do you hear me? Yeah. Okay, this is fine.
In spite of the micro is not doing anything.
Have I switched? Okay. Yeah. Okay. This is my title: Stabilization of the sometimes very low concentrations of Lyso-P in cancer patients. I think there's no need to explain Lyso-P, but what I have to explain is that Lyso-P is very important and, from my point of view, a very underestimated phospholipid metabolite as well as a signal molecule. You may all know that we have about 300 micromolar Lyso-P in our plasma when we are healthy. And since this Lyso-P has detergent-like properties and is therefore able to lyse red blood cells and therefore is able to induce hemolysis, this Lyso-P is mainly bound to albumin. Only a little bit of the Lyso-P is free. And since this equilibrium is very fast, all of this Lyso-P can be used by cells in different ways. And this is shown here. First of all, on the top, there is a transporter, well known since 15 years, MFSDA2A. This is a flipase, which brings in the Lyso-P into the cells. Once into the cell, the Lyso-P can be transferred, for example, in double-chained normal phospholipids. And these double-chained normal phospholipids can be used, for example, by lung cells to produce lung surfactants. This is important for breathing for us. Or, for example, the immune system. When the immune system has to proliferate, there is a big need of new phospholipids. And all these phospholipids can be generated through Lyso-P. And there are a lot more examples. Furthermore, Lyso-P is a very important signal molecule. There are a lot of different receptors for Lyso-P. Very important is this G protein-coupled receptor 119. And this receptor, this 119 receptor, is very important in a perfect glucose turnover. When you need a certain level of Lyso-P in your blood for a proper induction of insulin release, for example, a lot more examples. And then, also very important is the cleavage of Lyso-P outside the cells by a Lyso-P lipase, leaving behind the water-soluble alpha-glycerolphosphocholine plus a fatty acid. This fatty acid will be taken up by the cells as an energy source, and this can be seen in muscle and heart cells. And to give an example, about 70% of the heart energy is from fatty acids. Okay. Only examples. You might ask, a lot of jobs for Lyso-P. Is this 300 micromolar Lyso-P, which we have in our plasma? This is, for me, I have about 3.5 liters of plasma in my body. This is in my body, I have a total of 500 milligrams of Lyso-P in my blood. And is this 500 milligram of Lyso-P sufficient for all these processes? And the answer is it's not. But the good news is there is a lot more of Lyso-P in our body. And this Lyso-P is in deeper compartments. Most Lyso-P, and this is more than 95%, is in deeper compartments, meaning tissues and organs. And the equilibrium between the deeper and the plasma compartment is extremely fast. So you can regard both compartments as one compartment when it comes to measurements. The next slide shows you the phospholipid turnover in the body. The blue box, this is our body, all the deeper compartments combined. You see a blood vessel. In the blood vessel, we have 5% of the Lyso-P. The other 95% is in deeper compartments. When you calculate from 500 milligrams to come to the conclusion that in the total body in the deeper compartments there is about 20 grams. We are not sure about the exact values, therefore we usually say it's between 10 and 50 grams Lyso-P we have in our body. That's not all. Lyso-P, of course, has a half-life. The half-life of Lyso-Pc in the human body has never been determined, but we have data from mouse and squirrel monkey, and there it's about 2 hours. And when you take this value, you come to the estimation that the total turnover in the body is over 100 gram, much more than 100 gram per day. This is a lot. Glucose turnover is 200 gram per day. And it's isocaloric, this 100 gram of Lyso-Pc and the glucose. Yeah, but this is necessary because you have so many Lyso-Pc consumers, immune system, kidneys, muscle, heart, and so on. And on the other side, you have Lyso-Pc producers. This is mainly the liver, this is at least what we believe, and the GI tract, because a lot of this Lyso-Pc comes by food in our body. And there is a balance between Lyso-Pc consumption and production, and this results in this equilibrium concentration of about 300 micromolar in our plasma. This is the situation, but what will happen when another player comes into the game? And I'm talking about metastatic cancer cells. And we know from own studies that metastatic cancer cells have the ability to cleave Lyso-Pc extremely rapidly outside the cells. This is extracellularly. And this is from one of our papers. We tested 10 different metastatic cancer cell lines. We put in the cell culture dish 450 micromolar of Lyso-Pc and bound to albumin, of course. And and this is removal of Lyso-Pc. And you can see almost all cells completely remove the Lyso-Pc within 2 to 3 days. At the same time, there is an accumulation of triglycerides in the cells because the fatty acids were taken up by the tumor cells, and you can see the increase in triglycerides over time. And there are also pictures from microscopy studies. At the top, this is the control. Below is the Lyso-Pc-treated cells. This is a B16F10 cell, and we stain these cells with body pie. Then you can see the lipid droplets, and you clearly can see the thousands of lipid droplets which are in the cytosol. And calculated from all these data, or estimated, let me say, estimated from all this data, we came to the conclusion that 1 gram of tumor tissue is able to remove 1 gram of Lyso-Pc per day. This is a lot, meaning that even a small to medium-sized tumor is able to remove more than 50 gram of the Lyso-Pc. And the total turnover is more than 100 gram, therefore it's a big portion of this Lyso-Pc which goes to the tumor cells. And and therefore we expected extremely low Lyso-Pc levels in cancer patients. This is exactly what we found. We tested different types of cancers, different cancer patients. Here are the results. The mean Lyso-Pc levels are plus minus 200 micromolar. This is 1/3 less than in healthy persons. But what you can also see is that the standard deviations are extremely high, meaning that we usually have patients which almost have no reduction in Lyso-Pc, but we also have patients with extremely low levels. And and as a summary, I can say the more ill a patient is, the lower the Lyso-Pc level in this patient. Lyso-Pc has been discussed as a tumor progression marker. Here you have the very high Lyso-Pc consumption, the reduced Lyso-Pc in the body. And then the question, is this a problem? Yes, it is a problem. For example, in the lung, when there is not enough lung surfactant, you will have problems with breathing. This is what we see in our cancer patients. Most cancer patients have problems with glucose utilization. Most cancer patients are weak. The advanced cancer patients are weak. Might be in part due to the fact that there is only a pure muscle fatty acid supply, low Lyso-Pc levels. Most of our cancer patients have a lack of drive, lack of concentration, might be due to the fact that there is only a reduced Lyso-Pc uptake by the brain. And very important, most patients, advanced patients, have immune suppression, and this is a real problem when it comes to cancer immune therapy. Okay, I have to speed up a little bit. Therefore, we thought it might be a good idea to increase or stabilize lyso-Pc in these patients, and therefore we need a lot. Lyso-Pc, we think the only way is to do it by infusion. But there are a lot of people coming and said, okay, this is a problem. Lyso-Pc might cause hemolysis. You can't do this, but we can. The idea is shown here. We infuse Lyso-Pc into the plasma, and once into the plasma, Lyso-Pc can bind to albumin. From the albumin, it rapidly goes into deeper compartments. Then the albumin is free again and can take up the next Lyso-Pc. But you have to do it slowly. We tested this. This is our result. Here the yellow line, this is our hemolytic threshold. This is defined as 20% hemolysis values from in vitro studies. The red X is the basal level in these rats we tested. This is higher than in humans, a little bit less than 900 micromolar. And the black curve, this is a theoretical curve for Lyso-Pc increase in plasma, assuming only a plasma compartment, which is not true. But if you had only plasma compartment, the values will go up when you give 140 millimolar to over 10,000 micromolar. The blue curve is the curve we expected. This is a theoretical LPC increase in plasma, assuming an LPC distribution between plasma 5% and deeper compartments 95%. But this is not what we found. We found the green line. And this was surprising. There was a reduction in Lyso-Pc after we gave this Lyso-Pc of about 1/3, and after that it was stable. But what you can see is that the concentration of Lyso-Pc in the plasma, even we gave a huge amount of Lyso-Pc, is low, and therefore there was absolutely no hemolysis. And this can be illustrated. This was a pre-experiment, but good for illustration. On the left, we injected Lyso-Pc into rats rapidly. It took 30 seconds, and you clearly can see hemolysis. And on the right, we did it over a longer time, 3 minutes, and there is absolutely no hemolysis. But this is only for visualization. Yeah, let me come to my summary. There's absolutely no hemolysis during or after slow infusion of very high amounts of Lyso-Pc. Therefore, the theory is true. We again proved that there is a rapid distribution of Lyso-Pc between plasma and deeper compartments. We saw that, at least in rats, the deeper compartments are bigger than 95% for the Lyso-Pc, and you saw the drop in the Lyso-Pc. This is very interesting. There must be an additional regulation mechanism which controls the Lyso-Pc, maybe something similar to glucose level control or something like that, but we had no idea yet what it is. Yeah, I have to thank my friend Gerd Fricker and our PhD student Rebecca, who did a little bit of this work, the last experiments. Then there is a long list of PhD students, bachelor and master students worked on this project over the last years. I have to thank Phospholipid Research Center for funding this project. I have to thank my company Hettich to give me the time to do all this research. Kerstin Diehl Stiftung supported our projects. Then thanks to University of Freiburg, thanks to University of Heidelberg, and thanks to you for listening.
Thank you, Ulrich, for this beautiful talk about the Lyso-Pc and the relation to cancer patients. And thank you also for keeping the time so nicely. And we will have some time for discussion. Are there questions? Yes, over there.
Yeah, thank you for your very interesting talk. I see that you infuse these rats, I think, with mice cells. But have you also considered just binding it to albumin?
Yes, of course, we considered it.
Yes,
yeah, it's probably a good way because most cancer patients also suffered from a reduced albumin level.
Yes. Okay. And since you don't really see an increase in in PC levels, could it be that the micelles do not break down and they're just stable?
The Lyso-PC is in the red. This is clear.
Okay.
And,
yeah. Okay.
We believe that there is this activity on the surface of more or less every cell. Every cell has the possibility to break down Lyso-PC. And there might be a certain affinity to Lyso-PC. And if this affinity, if you have a higher concentration, then the excess of Lyso-PC will be degraded. This is a safety mechanism, we believe, to prevent the body from too high levels of Lyso-PC, because when the levels are too high, you will see hemolysis. And we did our experiments with young rat, and young rats have a very high Lyso-PC level, maybe close to this upper limit. This is a typical phenomenon we see in young individuals, rats, mice. When these individuals are young, the levels are much, much higher because there is a need for new formation of body compartments, things like that.
All right, thank you very much.
Hi. I just wonder if the effects will be different with other types of fatty acid change in the PC than you're used?
No, they are different. There are differences. What we only saw this in vitro.
Was it 16-0 or 18-0?
Yeah, for this experiment we used the physiological mix of 16 and 18.
Okay, one short question.
Very short question. Are there any approved products in the market with Lyso-PC?
Sorry?
Are there any approved products in the market with Lyso-PC?
No.
Short answer, no. Okay, we have 1 minute. Do you have a question? No. Okay, that's right.
But it is very unusual.
Okay, one last question over there.
Okay.
Have you ever tried imaging of the Lyso-PC with the radioactive tracers and see how the different?
There are older studies from the 70s. But this is true. This was a good time for Lyso-PC research. Then we forgot a little bit about Lyso-PC, and now it comes up again. There are studies, and they labeled the fatty acids and the head groups as well, and you can see the accumulation is different. You have, for example, a huge accumulation in kidneys, in lung, but there the both markers appear, meaning that you have the Lyso-PC as a phospholipid in these tissues. In other tissues like muscle or heart, you only can see the fatty acid.
How about in cancer? Is there like a cancer?
No data about cancer and labeling.